Welcome to the Natural Growing Through Biology podcast. This is our podcast series. I'm Steve.
And I'm Dennis.
And today we're going to talk a bit about the Phyllosphere and what's going on in the Phyllosphere, or the foliage of the plant, and how some of the microbiome associated with that can do some pretty cool things. So that's it.
You know, and it's really exciting, and I don't know that we've ever said this before, but anytime we're looking at foliar nutrition, the phyllosphere of the plant, trying to feed those plants, enhance root development, we always say, test, don't guess.
We do.
You know, it comes down to the idea of, we need to know what is in our soil, what is being made available to the plant, what deficiencies are there based off of nutrients within that soil environment but not made available because we don't have that microbial community yet built to release that nutrition within that soil environment. And part of that comes down, as we'll talk about later, is that foliar application, how this all functions together, which is really kind of exciting, that I'm excited about. And then we also have to use the plant sap analysis or leaf extract analysis to give us that information of what exactly does that plant need, and what things can we use to enhance overall plant health, fruit, size, quality, and all of those things. So
and figure out what sort of deficiencies you have going on, as well as the toxicities, those excesses, and those can be extremely detrimental to what you're trying to accomplish, because you can be putting a whole lot of work into phosphorus or into nitrogen or into zinc or something, but you've got another antagonistic element that's blocking everything you're trying to do. So you've really got to focus on that part usually first.
Yeah, and a lot of times we see that. I mean, sodium excesses, how that relates,
Chloride,
aluminum.
Yeah,
all of the things that we talk about. So when foliar feeding, I mean, we kind of talked about, so why?
Yeah.
Why would you foliar feed? As we said, a correction of nutrient deficiencies that are noticed, to correct an imbalance or an excess.
Yep, makes sense.
And for poor root development. A lot of times out when I'm talking to growers, we see poor root development or damaged roots, and they're not able to take up that nutrition from that soil environment. I mean, this could be from disease, nematodes, insect damage, whatever the case might be, drought. All of these scenarios would be when you could utilize foliar to get that plant health, because the plant's not functioning as designed. And any time with new planting, where we put that new plant into the ground, we've obviously dipped it in mycogenesis. It's got a great start.
Oh, yeah.
But what we need to do now is do a little bit of foliar nutrition to kind of speed that process up and ensure that healthy root development within that soil environment, because all of these things are complementary to a fertilizer program or a soil applied foliar nutrition program. They're complementary to that. So it's really kind of important that we think about it that way. Is foliar nutrition goes well beyond, we'll talk about this later, Steve, but a lot of times people say, well, I don't put out very much. How can it make that big of a plant response? And I think that's some of the stuff we're going to kind of tie all together today for the grower to understand how important a small application of foliar nutrition can make in overall plant and soil health.
Oh, definitely. And no, you're absolutely right. And like with our products, for example, it's a small amount of product, but that's because we're trying to kind of we're sowing the seeds of these organisms so they can be on the leaf surface, so they can be utilized by the plant. Because like we say oftentimes when we're talking about soil in the rhizosphere, the plant can only use the tools it has access to. If those organisms aren't present, it can't be used. The plant has no way of just magically material. It doesn't have a teleporter. I mean, Star Trek technology would be really cool, but plants don't have it. I mean, we don't have it. Plants don't have it yet.
Yet.
Yeah.
But, and sometimes we look at, when I talk about foliar nutrition in addition to, root development, but we have to also look at that when, California this year, when we have the excessive flooding and we have saturated soils or drought conditions that we're seeing in the Midwest, stress conditions, we talk about hail in, Kansas, Nebraska, North and South Dakota, these hailstorms that are going through, especially when they go through early, a lot of the stress reduction can be mitigated also with foliar nutrition, is one of the things that we kind of want to take a look at. In each one of these instances, again, test, don't guess, have that information so you can understand how to utilize it, but can make a huge difference on how that plant heals itself.
Oh, definitely.
So when we start to talk about that, we always talk about feeding biology. We always talk about when we're working in that rhizosphere to make sure we have some kelp, make sure we have some fish, make sure we have some humates, make sure maybe a little molasses is in there. Carbon. But really the best way to feed biology is a lot of times with root exudates. Yeah. Pumping up those exudates, get those plant exudates going down, because sugar, carbon, all those things we're talking about, the communication that you've talked about a lot, from the standpoint of that, all comes down to a healthy plant. When we start to talk about how much sugar does that plant share, well, when it's stressed, it's not sharing a whole lot of its sugar because it's going into growth and reproduction. Yeah. So when we start to talk about that, foliar nutrition is a great way to enhance root exudates within that soil profile.
Well, and that you mentioned this quote up here, poor root development, using foliar feeding to help with poor root development. That's exactly it. I mean, if we're utilizing these organisms, and a great way to test a Brix measurement, if we go out to our fields, and I know Brix is a bit of an art form. It takes some work, it takes some time, you have to get used to doing it, how to do it, you have to be able to replicate what you've done. So it isn't just a simple walk out, grab one and done, and that's all you have to do. You have to kind of continually do it, and you have to get to know it. But once you have that tool, and you can trust that tool, you can start to do things like see when my plant would need some of these foliars, and then you can test after you spray to see if that foliar really helped your plant. If you see the Brix go up a few points after a foliar, that's a pretty strong indication that you have been able to increase photosynthesis with that nutrition. And it's not necessarily saying this product is bad, that product's bad, this one's good, that one's whatever it is. That has more to do with timing and what the plant needs right then. You can have a product that works great 95% of the time, but you might have a situation where it's not quite the right tool. And that's a lot of times where the test don't guess comes in. And then once we do that spray, we can utilize Brix. And when we see that Brix go up, the beauty of that is we know now that thing you're talking about, those exudates, we know that they have increased. That plant now has a bit more sugar. It has a bit more energy. It's a resource. I mean, you can think about that sugar like a currency. Everything runs on energy, and that sugar is the currency of the plant. And it's got to pay those microbes. So like you were saying, when it's dehydrated, when it's not photosynthesizing, well, it's been cloudy for a few days, you can start to see some of that sugar drop, and those can be good situations and times to throw a little bit of sugar out with your foliar spray. Yeah. Cloudy conditions are a great time, and that helps feed the microbes.
If you're not comfortable using a refractometer and pulling the Brix and you don't have that information, again, that plant sap analysis, leaf extract analysis will have that information for you. It'll talk about sugars basically in the plant.
It does.
And so for those that, as you're building that tool, if you want to start using a refractometer and measuring your own Brix, as you're building that and getting an understanding of what nutrition to apply as a foliar application, again, that plant sap analysis will give you that information, and then you can start making a correlation between the plant sap and the application that you make and checking the Brix readings to get an understanding of how things function on your own farm.
Oh, absolutely. No, the refractometer on field, that gets you the tests in between when you've sent it off.
Correct.
Because no, that does not take the place of. You still need to continue doing that testing, because otherwise you are...
You're guessing.
Oh, I set it up.
See, you did. Thank you. And good thing you had those flashcards for me so that I could look at and read those.
We'll have to bring those next presentation, like the applause ones that you hold up, so people remember, oh yeah, no, that was good, guys. Laugh now.
Well, and when we start to talk about that, laughing now, so often when we start to talk about foliar nutrition, we'll talk specifically about nitrogen, because that's one that's out there a lot that we try and get growers to cut back on. A lot of times there's excessive nitrogen being used in that soil environment. And we look at that and we start to say, okay, reduce your nitrogen and go out there with a pound of soy protein nitrogen, and go out with these microbes that fix nitrogen on the Phyllosphere of the plant. We start to talk about that, and it's just amazing the research that is out there now that talks about how these nitrogen fixers can basically supply nutrition and nitrogen specifically to that plant.
Oh, absolutely. So this is research from 2016. The name of the article is Assessing Inoculation Methods of Maize and Wheat with Azospirillum braziliensis. And this is from A&B Express 2016. So in-field experiments, inoculation with Azospirillum braziliensis, or A. braziliensis, allowed for a 25% reduction in the need for N fertilizers. The analysis of results from a large number of field trials with various non-legume crops conducted worldwide over 20 years under different soil conditions and weather conditions that demonstrated that yield increases of up to 30% could be obtained.
And it's amazing because you talk about different soil conditions, different plants, different crops, all of these scenarios. And this is one specific strain of a nitrogen fixer, which is really amazing when we start to think about that, because we know there's more than just one.
Oh, absolutely. And,
well, I've heard that anyway.
Well, there are a couple out there that can be used and should be used and can do amazing things. This research was basically done, though, kind of just to prove that, these guys can do the job. I mean, people ask the question kind of all the time, are these going to do anything? Can they make a difference in my field? This was in field conditions, and this was over a long period of time. And again, it's just one organism. But what these researchers are finding is Azospirillum braziliensis, it's an endophyte. So this organism has the capacity of being sprayed on the tissue and then moving inside of the plant. And in that environment, it can fix even more nitrogen. So it has a very, very strong ability to do that. And that reminds me and makes me want to talk a little bit about some of Dr. White's research. And he was looking at what's going on those trichomes. We think about the trichomes, and we think they're just little hairs, right? They don't do very much. They're just little hairs on there. I talked about them in a previous presentation, a weird one. I was talking about wild tobacco and the trichomes and how the plant will modify its trichomes to feed the organism, to cause it to metabolize that exudate or that material in the trichome and make it stinky so that predators will find it. Okay, so now we're going to talk about something different. So if that sounds interesting, check out a previous one. So in this research, Dr. White was looking at hemp glandular trichomes. And we think about those, we think about trichomes, we think about hemp or cannabis. That's where the essential oils are. That's where we find all, those cannabinoids, those materials that people are excited about, all those essential oils, all those plant secondary metabolites. So if you're trying to increase the oil content or the essential oil content or the plant secondary metabolite content, these trichomes are a source for some of that. And in this research, he's finding that a lot of those materials, what they're actually doing is they're oxygen scavenging molecules. We talked before about nitrogen fixation, how that nitrogenase enzyme is extremely sensitive to oxygen. If you're on the tissue of a leaf, there's a lot of oxygen. So having this zone, having this specific space where you have the plant producing a molecule to protect you and protect that function, it's ideal. So in these sites, we're seeing a lot of nitrogen fixation happening if we have the organisms to do the job. And we'll talk a little bit later, there is research, like Dennis was saying, about sometimes our fertility programs can affect the diversity, the alpha diversity of the different organisms. And we can decrease some of those organisms in the plant, which doesn't help. And it's not just the glandular trichomes. We also see the nitrogen fixation occurring in these hair-like trichomes. So if we start thinking about these trichomes as a site for nitrogen fixation, that kind of changes your view on them. That gives you a different perspective into how important all of this surface area of a leaf can be. And that's another indication we start talking about as we see leaf size increase. More leaf size, more photosynthesis. But that also gives us a larger surface area for some of these biological processes to be going on, which is just incredible, and it's very powerful.
Well, and if you start to think about that based on the phyllosphere of the plant, and we often talk about solar panels based off of photosynthesis. And you know, I think Bruce had mentioned, and we talk about this a lot, and I'm going to say it again, everything is interconnected. Every bit, every piece of this plant, these microbes, the soil, is all connected. And when we start to think about that, if we have bigger leaf structure, yeah, we have more photosynthesis, but we have more organisms on that phyllosphere able to function in this way, producing nitrogen, making nutrition available, and just some of the other things we'll talk about here later in the presentation. If we start to think about that, everything we can do to increase leaf size, leaf health, leaf quality. So we're always so focused on, when we look at a deficiency within that plant, how that relates directly to yield. But it goes well beyond that.
Oh, absolutely.
And when we start to talk about defense mechanisms and nutrient availability and water retention, all of these things are kind of what we want to touch a little bit, and why foliar nutrition is so important, because it can make such a huge impact on the plant.
Yeah, and like we already talked about, it's not just on the leaves, it translates throughout the entire plant. Another bit of research kind of wanted to talk about, and I have this slide labeled Phyllosphere Friends. That's a pretty fun one. This is from, and I'm going to flip over, Phyllosphere Microbiome, Diversity and Functions, and this is from Microbiological Research from 2022 is where this article is from. So this is looking at some of the different organisms that are associated with the Phyllosphere. Not all of them are friends. A lot of these are pathogenic microbes. These are disease-causing, and it's plant disease, but it's not just plant disease. We can also see where some human diseases can be transported on these. I mean, I haven't heard it in a couple years, but it seems like every few years you hear about those E. coli outbreaks from this crop, or I'm not going to name crop names, I don't want to shame them, but you know, sometimes it happens, and sometimes it's, you know, a little bit of unhealthy cattle manure, something like that gets spread, gets too close, or I'm not going to say how or where it comes from, but
environment.
Yes, we'll say the environment. So that's the bad side. Then there's also these non-pathogenic, and they're beneficial organisms. So they have different ecological roles, and they have different roles in the plant. So the ecological roles, we can be talking about climate regulation. We've talked about abiotic stress. We've talked about it mostly in association with those plant growth promoting rhizobacteria, but we're seeing in the research that these organisms on the phyllosphere are also playing a pretty critical role in how that plant responds to abiotic stress. And go ahead.
Well, and I was going to, you know, I'll bet it was 20 years ago, I was in Colorado and Bruce came out for a visit, and we were in a huge drought stress scenario. We were working with a corn grower out there, and it was interesting because he ran out of irrigation water because of the drought scenario. His conventional field was melting down, and our biological field was still doing pretty well, and Bruce came out and said we wanted to do a foliar on that. And so we talked the grower into flying a plane, doing a foliar application, and we added one thing, which was PZ1000, which was a foliar microbe and a very light nutrition package. Grower wanted to come out with more nitrogen and all of these things, and Bruce is like, no,
just
this
try
one
it.
thing.
Just try it.
Just this one thing. And it was pretty amazing because this corn actually finished very well. He used it all for his feed, for his cattle. This biological program exceeded the yield of his conventional program. The yield actually went up based on the field scout after this foliar application. So the plant continued to grow, but it's what you're talking about. It was reducing that stress.
Yes.
That's Bruce's whole intention here, was to give that a couple more weeks to finish. That's all he wanted to do. And sometimes when we start to think about it that way,
that's all it takes. But that finish, getting a few more of the kernels filled, and that makes a huge difference to your yield, or sometimes with different crops to your test weight. Or we're talking about like potatoes, that, you know, a lot of times you see the potatoes grow and then they're done. They're down in the field. When we give them more time and we're photosynthesizing longer, it's like we've got that bike pump, and we get to have a few more pumps to fill it up, and that can make a huge difference. Let's see, phytoremediation and pollution control. So we're actually seeing some of these organisms, like we've seen and we talked about in the soil, some of these organisms are taking care of the pollution that people are putting out. Not great. They're actually tying it up. Some of them are, depending on the molecule, they're decomposing and breaking some of that pollution down, as well as sequestering some of it. But also, these organisms, when we have them in adequate quantities, they help reduce the methane production from the plant. So we're having less greenhouse gas. Because, yeah, plants do produce small amounts. They do respire. They have respiration going on, just like you and I do, just like cows do. They are also producing small amounts. But these organisms, they utilize that nitrogen. They grab that nitrogen, they utilize it, so that we're not having as much moving off into the air. So some of the nitric oxide, and they're utilizing the carbon from the methane. That's CH4 molecule. That's just carbon. They're happy to grab it and utilize it. So they're reducing some of those greenhouse gases that are coming off the plant. I think that's pretty cool. So having this biological system, it stacks, it compounds. Having more of them allows that plant to do more of the things that it naturally should.
And all of this is related as we start to go back to it. I mean, I've already forgotten, but we're talking about foliar nutrition and how that enhances the overall plant health.
Oh,
because
yeah.
that's really what this whole conversation comes down to, is all of these different things that are happening on the phyllosphere of that plant in regard to biological communities, regulation, nutrient uptake, and really how that affects every part of not only plant health but also soil health.
Oh, because we've talked about that translation. It's a back and forth. It's an up and down. And I've got a pretty good picture coming up that shows more of that. The role in plants: nutrient acquisition, pathogen defense, production of growth hormones, and stress tolerance. We've talked about a lot of those. Most of those we've talked about when it comes to plant growth promoting rhizobacteria. And it's weird. It's like we keep saying these organisms, they have similar functions above as they do below. The plant needs the same things, whether it's above the ground or below the ground. The below ground can be stressed. The above ground can be stressed. You just showed me some pictures of cherries, and they needed water.
Yeah.
They needed water pretty bad. There are stresses that are going on associated with the above ground parts. So these organisms, through the ACC deaminase and through those similar pathways, they're helping that plant. They're helping that plant with nutrient acquisition, with potassium. We have potassium mobilizers, and I'll share the quote of that here in a second. But we have those organisms that are helping that potassium move into the plant from the phyllosphere that helps the stomata open and close. So we have that coming from both directions, with these organisms from the roots helping, as well as these organisms physically on the leaf, helping that stomata open and close to preserve the water, producing antioxidants, producing these materials that help reduce reactive oxygen species when the plant is stressed, producing those phytohormones to help dictate and regulate how the plant is growing. As soon as we start shifting over to that ethylene, we start to shut down. We go to senescence. We have that plant slowing and shutting down. If the microbes are sending out more indolacetic acid, citric acid, more of the gibberellins, more of these growth hormones, and the plant continues to grow. At the same time, these organisms are degrading and taking up some of that material and shutting some of that down.
Well, and so often when we talk about yield and increasing yield, you know what I've always said is basically increasing yield is managing stress. Everything we can do to reduce stress in that plant increases yield. And that's from the point of germination. We've kind of had that conversation before.
The idea of genetic potential.
Yeah, and we just build upon that. And that really is what a lot of this comes down to. And a lot of times, visually, you may not even see the stresses in the plant. By the time they're expressing themselves, there's significant amount of stress in that plant. And that goes back to the idea of these foliar applications and basically these plant sap analysis and all these things. Before we even see that stress in the plant, we see the yellowing leaves, we see the insects, we see the disease. Long before that, we're already compromising yield. And that's why it is so important that we're proactive on these foliar applications rather than reactive by the time we finally see it.
Oh, absolutely.
Okay. Well, this has been a lot of fun. I want to actually Appreciate everyone that hung on through this, listening to Steve and I ramble.
Having fun.
Having fun. Again, this is Dennis. Thanks everybody for joining us.
Yeah, and I'm Steve. Thank you for joining us while we talk about the Phyllosphere and the phytobiome, the microbiome of the plant's leaves on our podcast.